Welcome to this LASER Zurich event about how and why artists and scientists are conducting research into the future production of food and medicine. Using chicory components as an example, the concepts of wild plants, cultivated plants, and in vitro plants will be explored in this session. It will feature 6 presentations by media artists and biochemist researchers who are involved in our current Art and Science Node Artist in Residence program called CHIC. The presentations will be followed by a discussion about the future of food and medicine. Chicory is a native European plant with beautiful blue flowers. It has also been cultivated for coffee and medicine. Currently, some commercial cultivation of chicory takes place to produce the food fiber inulin. The next part features a new documentary film excerpt about inulin production and how biochemists are exploring its potentials as a low-calorie sweetener, a fat replacer, and even as a probiotic for the health of the colon.
Hello. The best way for me to participate in the seminar is to show a segment of the film I recently directed at Census in Holland about the production of inulin for chicory. We were amazed at the different processes to gain this probiotic fiber, and I imagine you will be as surprised as we were about such an interesting level of cultivation.
Chicory is a wild plant. It is known in Germany as die Wegwarte, or the plant that waits along the European country walkways. We were told that it's native to Europe. And then we found it right on our daily walk in the middle of Zurich on the river. Farmers say that if you plant it in the field, it is much stronger than other crops, and it improves the biodiversity of the soil. But chicory roots also have healthy ingredients for us. Today, farmers' cooperatives like Census in Holland are growing chicory for its high-grade fiber and sugar substitute potentials. But it has only been cultivated for the last 25 years. Scientists tell us that chicory contains a type of dietary fiber called inulin. Their research has linked it to several health benefits, such as improving digestive health or helping to control sugar diabetes.
Oh, I'm interested. They're all different sizes, but they're huge.
That is indeed the objective. I mean, we want to have as much mass as possible from an acre of land, because we are making an ingredient out of it. So the more the mass, the more ingredients we get of the land.
The chicory root is a very robust crop, so it doesn't need much fertilization, doesn't need much pest control. It is slowly growing. That means that certainly at the start, the farmer really has to pay attention to weed control. So this is to facilitate sowing, and this is really the clean seed. And here you see different versions of pillage. Which also contains some nutrients that the seed needs for getting out of the ground. So we work with 500 farmers approximately. They sow the seeds from March until late April, and from there on it's basically their responsibility. The chicory grows until we start the harvest.
In September, we went with Matthew to one of these farms.
Started about 6:30.
Harvesting already, 6:30.
Yeah.
Chicory roots are grown in the Dutch fields and are harvested from September until November.
So when will you be finished? What do you think?
What time? Or
Yeah, what time?
This about 9 or 10 tonight.
You'll be finished.
Yeah, I think.
Oh, it's going to be a long day, huh?
Yeah.
The farmer tells us that he puts the leaves back into the soil. We also learned that chicory leaves help to retain the moisture and feed the good bacteria, fungi, and other naturally occurring organisms in the soil. This results in a natural release of nutrients into their roots.
It has been a good wet season.
Yeah.
If it's a dry season, last year we had a very dry season, then we get very long roots. So then, because they're looking for water.
Amazing, huh?
Yeah.
They're really good sized roots, aren't they?
Yeah, these are good size roots indeed.
Yeah, very
The
good.
factory will be very happy.
At Senses, we were surprised how complex it is to extract inulin from the chicory roots. Inulin is the main prebiotic dietary fiber in the chicory root. Here we follow the processes of delivery, sorting, washing, slicing, and extraction with hot water and additional purification processes to produce inulin powder or oligofructose syrup.
So they are shoveled into the pit and then brought by a conveyor belt into the factory. First it's checked on metals, then they are washed, and then they are undone from weeds that might still be present, stones, etc. And then they are grated. The roots will then be immersed in warm water, like about 60 centigrades, and then they are brought to our extractor, where inulin is extracted from the roots with water of approximately 70 centigrade. And to have inulin at the end, we need several purification steps to have pure inulin in water. First we start with the demineralization step. So there we get rid of the minerals that are in the plant. Then we have an active carbon step where we get rid of the terpenes. So after this step we have pure inulin in water, which we can taste in the factory.
Yeah. Cheers. Yeah. Tastes good, doesn't it? Now you?
So after all these purification steps,
We're going this way.
then we evaporate the water until a high enough level, and then we spray dry it to yield only the inulin powder, which we package in bags and then can distribute to our customers.
The leftover products and minerals from these processes are not wasted.
After extraction, we did not only extract the inulin, we basically extracted everything that dissolves at 70 centigrade hot water. So also the minerals that are in the plant, also the polyphenols that are in the plant, and of course also the terpenes that are in the plant. So all the water that comes from the factory is treated in a wastewater system. The aerobic part, there it is cleaned and then spilled on the surface water. The anaerobic part is making biogas. So 25% of the gas usage that we have in our plant is our own biogas. The different qualities of inulin that we produce have different qualities in the end products. The longer chain products, they have a use as a texturizer. They provide a mouthfeel that is similar to a fatty taste, so therefore can replace fat. The shorter chains have a slightly sweet taste, and therefore are very well used for sugar replacement. The challenge with our current process is that it is fully optimized for yielding inulin, but it would be much better if I have a process that gets the terpenes out in the most efficient way and the inulin out in the most efficient way.
Thank you, Marilla. And now we move on to our next speaker, Dr. Ingrid van der Meer. Ingrid van der Meer studied at the Free University in Amsterdam on the regulation of plant gene expression. She has more than 25 years' experience in plant biotechnology. She currently is head of bioscience at Wageningen University and Research. She coordinated the research project focusing on inulin and rubber biosynthesis in Russian dandelions, and works already for more than 20 years on inulin biosynthesis and chicory.
In this presentation, I would like to tell you something about the research that we are performing within the EU CHIC project. EU CHIC is focusing on chicory as a multi-purpose crop. And my part of the research is focusing on inulin. I will explain a bit more about chicory, how it started as a wild plant to be used as a crop, and I will focus specifically on improving the quality of the inulin. Okay, chicory as a wild plant, a bit about the history. It's native originally to Western Asia, North Africa and Europe, but at the moment you can see it in the wild. It's very common also in North America, China and Australia. And the history of humans using chicory as for food goes back already to ancient Egypt, and it has been described in ancient Rome to be used as a vegetable in a dish where the young chicory sprouts are used as a vegetable. Well, the first real cultivation started in the 17th century, so not taking it from the wild. But then people started to cultivate it and select. Specifically, in first instance, the roots were used. So the root chicory, where they roasted the roots, and that was used as a substitute for the coffee or to add it to the coffee because of the nice bitter taste. But also people have been selecting more specifically from this same species, the leaf chicory, where it's now known as, for instance, radicchio or the Belgian endive, you know, these white endives, or Puntarella or Catalona chicory. So the focus on using chicory roots specifically for the production of inulin, that started about 50 years ago, 1970. So then people start to cultivate and also to breed specifically for root chicory for the production of inulin. So a bit more about this inulin. And then you see these really big plants. So first, crossing and selection, that's actually breeding. So you pick a specific plant that has a nice phenotype, like big roots, and you cross it with another plant, and then within the progenies, you select the ones that have bigger roots. And that resulted eventually in what we call the root chicory, which you can see here. So this is what you see in the wild, and this is what you can see now. And why did people want to have these big roots? Because the plant produces inulin in the roots. So for the plant, inulin is a storage carbohydrate. It's actually energy that the plant can use. It's a plant fiber, and it can use this energy when, for instance, after winter, the plant needs to grow new leaves. But we also can use this inulin, and we are using it. So it is extracted from these big roots, and by us, it's now used, and already for 50 years, as what we call a low-calorie sweetener. So it has a sweet taste. It's a carbohydrate, but we, with our enzymes, cannot degrade it. It can be degraded or used by our gut microbiome. It's also used as a fat replacer because it has a specific mouth feeling. And since it's used by our gut microbes, and especially by the beneficial gut microbes, it's more and more used as what we call a prebiotic, a dietary fiber. And that's what you can see here. And what's, I think, special for chicory and inulin is that it also has 2 approved health claims by the European Food Safety Authority. So chicory inulin improves stool, maybe not that fancy, but it also improves the gut health. So it's really a very nice, beneficial product from this plant, the root chicory. And it's especially this root chicory that we are focusing on within our EU CHIC project, because we want to use it as a multi-purpose crop, and we also want to improve the inulin, the quality of the inulin in the chicory. And that's actually what I'm focusing on. So we want to improve the inulin quality, and we want to do this by using what you call new plant breeding techniques. You will hear more about it in the next scientific presentation by Katja. And we are using this technique to improve the quality of inulin. We want to reduce the hindering presence of bitter compounds because it hinders the extraction of inulin. We also want to have less small sugars like the sucrose, glucose, and fructose. These are not really beneficial for your gut health. And we want to have really longer inulin molecules because these are the more healthy ones, let's say. So within this project, in order to use this new plant breeding technique, we start with single plant cells. We make the mutation. We can generate from these plant cells whole new plants that have the mutation in them. And that's what we end up with, the whole new plants that can be used for seed setting and to be used as a new crop. Well, how do we want to do this to get this high quality inulin? By, as I just explained, use this new plant breeding technique, also called CRISPR-Cas9. And we want to use this technique in which we very specifically can make mutations that we want to use in order to block what we call the breakdown of inulin that normally takes place in autumn. Just before the farmer starts harvesting the plants and want to isolate the inulin, the plant under this cold stress starts to break down the inulin. And that's something that you do not want. Well, this is actually what we did. We did try to make these mutations, and the modified plants were then afterwards analyzed what we call autumn conditions. So that's what you can see here to the right. So the plants are stored at 4 degrees, and then afterwards we could harvest the roots, and that's what you see to the left, and analyze the inulin composition of these roots. Indeed, we could produce chicory which have less small sugars that are negative, that really have a higher inulin quality and have much longer inulin molecules. And that's something that you can see within this chromatogram, where each peak shows a different length of the molecule of inulin. And it can go up to quite a long inulin molecule. So as a conclusion, we could show that new plant breeding techniques can be used to induce specific mutations in the plant genome. And this is also a much faster method than breeding, and also much faster than the traditional mutagenesis that is used in breeding. And we were able to develop chicory plants with improved quality of inulin. And actually, we now have a cheaper method that can be used for the extraction. So there are on 2 ways it's beneficial for consumers, in both ways, because it can be extracted in a cheaper way, but also it has higher quality. Well, this is actually what I wanted to explain to you, and thank you very much for your attention.
Thank you, Ingrid and Marilla. Now we will move on to the next section of our LASER Zurich event.
Here we present a new interactive artwork and current scientific research on the benefits and molecular structures of wild root plants. Our first speaker is artist, Professor Dr. Jill Scott, presenting her Aftertaste, a molecular orchestra. Jill Scott is an artist, teacher, and context provider with 40 years of experience in the unique transdisciplinary field of art, science, and technology interactions. Her research spans neuroscience, electronics, ecology, sociology, sculpture, performative installation, and media art. Her interactive media work is focused on the human body, the social and physical impact of biotechnology on our bodies, and the health of our environment.
Hello, I'm going to talk about Aftertaste, the molecular orchestra. One thing that's interesting for me about all of this work with the CHIC project is that I started to think about a haptic media artwork that allows the audience to take on the role of air to transport molecules like this from the nose to the tongue. They use sound and touch to explore molecular activity, and they use the model of the olfactory bulbs to identify the molecules and the families they belong to. And they place the molecules into a 3D sculpture of the human tongue, which you see behind me, to explore the health benefits. So aftertaste is really about the sensation of smell and taste and how they combine to make flavor. So the sensation left after taste, smell and texture when they're no longer affecting the brain is called aftertaste. Hence the title. As we know, odorant molecules are transported through the olfactory bulb, then to the thalamus gland and on to the brain. And whereas compound molecules are transferred by the taste buds that send signals to also the thalamus gland to be recognized by the brain. So my methods of a media artist are to use embodiment, scale, and interaction. So in embodiment, I use video and sound to immerse the public into a special environment. In scale, I build models from microscopic images of the sensory organs that match a 5 nanometer scale of the human body. And then in interaction, I design the interaction to match and understand the relations between the molecules and human health. So what happens is we have a whole lot of different molecules in a plant like this. We have terpenes that are anti-inflammatory, antibacterial, antifungal. We have phenolics that are antioxidants, inulins that are related to gut health, nutrients and minerals that are really important for our growth and basic processes. So in the molecular orchestra, I try to have an instrumental orchestra, a body orchestra, and a natural family orchestra. Here's the first segment of the instrumental orchestra. And here's another segment of the body orchestra. And here's another idea about a natural family orchestra. Now I'm going to play a small film that shows how the audience interacts with aftertaste. Oh! for listening, and thank you to my team who helped me build this complex project called Aftertaste.
Thank you, Jill, for your fascinating presentation. And our next presentation is by Katarina Kankar, who is a senior scientist in Wageningen University and Research in the Netherlands, with 12 years of experience in terpene research. Her main focus is on terpene biochemical pathway discovery and metabolic engineering. And since 2007, she participated on several projects on plant cell wall engineering and secondary metabolism engineering at Wageningen.
My name is Katarina Zankar from Wageningen University and Research, and today I will tell you about chicory as a source of bioactive terpenes. Chicory plants belong to the plant family of Asteraceae, and they have a very long history of human use. The root chicory varieties are used for extraction of food fiber, inulin, and the leaf chicory varieties such as radicchio or wheatloaf are used as a vegetable. Now, the wild chicory also has a very long history of use as a medicinal plant or as a coffee surrogate. What all of these different looking chicory varieties have in common is that they're all tasting very bitter, and the bitterness of chicory is caused by the presence of small molecules called the terpenes. Terpenes are plant secondary metabolites, and although they are all made from this single building unit called the isoprenoid building unit, plants can make 40,000 different structures of terpenes. They are very important for plants in their communication with the environment, for example, for attraction or for defense. Some of them that are depicted here are also very interesting for human use as flavors, as fragrances, or even as medicines. Chicory terpenes belong to a special class of terpenes. Those are the sesquiterpene lactones. And what is typical for them is that they have a lactone ring, and that makes them very bioactive. In chicory, the terpenes are accumulated in this white substance that you see here on the cross section of the chicory root. This is the chicory latex, and the latex is then stored in tubes in the root of chicory. They're called laticifers, and you can see them nicely stained on the cross section of the chicory root here. Chicory terpenes are not only bitter, but they are also very bioactive, and they have been shown to have anti-worming activity in cattle, but they also have activities that are interesting for human use. For example, anti-inflammatory and antibacterial activities. In the past years, we have studied how the terpenes are being made in chicory, and we learned a lot about the genes and the enzymes that are involved in the pathway of chicory terpene biosynthesis. And we can use this knowledge if we make small modifications now in this pathway. We can either inactivate the terpene biosynthesis or modulate it to have a different terpene composition. The way that we do that is by new plant breeding techniques. We use the CRISPR-Cas9 approach. And in this case, we use the Cas9 protein to make small changes in chicory DNA, in its own DNA. And that's a much faster and much more precise breeding technique than the classical breeding. In practice, this looks so that we isolate single cells from the chicory leaf. You can see them here. We make a small genetic change in them, and in about 5 months, we can regrow chicory plants from a single cell. Using this approach, we obtained varieties of chicory that lack bitter terpenes. This is very important for inulin production. Inulin fibers are used as sweeteners, and the content of bitter terpenes is actually not desired in this sweet extract. Therefore, the bitter terpenes need to be removed during inulin processing, and this is quite a costly process at industrial scale. So to obtain chicory varieties lacking the bitter terpenes, we actually inactivated the first gene that is leading to terpene biosynthesis, And by that, we got varieties lacking the terpenes. And here you can see that 2 varieties that are shown here have nearly eliminated terpene content. By that, we can have a cheaper and more sustainable inulin extraction process. In the CHIC project, we also believe that terpenes are very interesting second products from Chicory besides the inulin. Therefore, we are doing further research on the Chicory terpene bioactivity. We are also working on Chicory varieties with enhanced and modified terpene composition. We are also studying the safety and environmental impacts of making new varieties by the new plant breeding techniques.
Thank you, Katja. And because we have a discussion at the end, we now move on to the next section of this LASER ZURICH event.
Biochemists in the CHIC 2020 Horizon Project use new breeding plant technologies to enhance the health benefits of inulin and to produce bioactive terpenes with medical applications. Our first presenter in this section is Dr. Suvi Hanneken. She's a senior scientist at the plant biology team of VTT, a leading multi-technological applied research organization in Finland. Her experience includes plant genetic engineering, modular cloning, plant genome editing, bioconversion, statistical experimental design, and as well the cryopreservation of plant cells.
Hello, I'm going to tell you briefly about the bioactivity toolbox that we are using in order to understand the health benefits of these modified chicories. So as said, we are using chicory as a multi-purpose crop, and our aim is to valorize the underutilized part of chicory for human health benefit. So we collect chicory, we extract inulin, which is the main dietary fiber in chicory, as we heard in the earlier talk already. And besides making improved inulin, we are also interested in valorizing the terpene fraction, which is currently not utilized at all and it is discarded. And we want to understand how we could upgrade the terpenes from chicory for different types of application purposes. So we have a toolbox for analyzing bioactivity in this project. So we are working with antimicrobial assessment, meaning antibacterial, antifungal, antibiofilm activities are measured. Then we are working with antioxidativity assessment. Then besides bioactivity, we are also interested in knowing whether these samples or extracts possess any toxicity to living organisms. And we have a platform for working that. Then we are working with anti-inflammatory assessment and also we mimic the human gut by using human gut models. And I will tell you a little bit more about those also. So antimicrobial assessment, we are really interested in chicory terpenes towards human harmful bacteria. So we want to understand whether we can somehow prevent the growth of different types of harmful bacteria and fungi. And we are also working with antibiotic resistant bacteria. So as you know, the antibiotic resistance is a huge global threat currently, what we are facing. And interestingly, we have shown that these chicory fractions, they possess activities also towards these resistant bacteria. Then besides harmful bacteria, we want to understand how these samples affect the beneficial bacteria. So for example, in human skin or human gut, we have a lot of different types of beneficial bacteria which are which are contributing to our health. And we want to make sure that those are not affected in a harmful way. And then we are also working with antibiofilm activity. So biofilm is the formation and that microbes need in order to survive and to reproduce. And if we can prevent the growth of the biofilm, then we can also reduce the survival of the harmful bacteria. Antioxidativity assessment, we are working with radical scavenging activity. So we are measuring the oxidation prevention. And besides human health, so of course, antioxidative activities are important to our health. But thinking about the possible products, this oxidation prevention activity might be interesting in that aspect also. And then toxicity. So we want to make sure that these highly active natural compound extracts or fractions, they don't possess major toxicity towards living organisms. And we are first screening these samples with photobacteria. So in the lower picture, you can see that the Vibrio fischeri, which is the bacteria which has this bioluminescence action, and this bioluminescence can be correlated to the toxicity. Then we are also working with freshwater crustaceans, so Daphnia magnas, and these are sort of a little bit more elaborated organisms, and they are very often, this Daphnia magna platform is used for measuring the wastewater toxicities, for example. But it has been shown that these little Daphnias, that the results coming out from that platform also correlates quite nicely to even higher organisms. So we can estimate the toxic effects also to other organisms in some extent. And then we are also working with human cells, so human cell lines in the laboratory environment. So for example, intestinal epithelial cells or some other types of cell lines. So we are exposing these cells to these compounds, and we want to understand how they are affected. And then finally, we are working with human gastrointestinal model. So we have a method where we can mimic the conditions in the human gut. So we are mimicking the conditions in the mouth, in stomach, and in upper and lower intestine. And we expose the samples to these different conditions, and then we follow the result. And we are especially interested in the colon fermentation. So for example, these modified inulins that are produced by the modified chicory. So we then measure the possible gas formation in the gut model, and the aim is to show that this modified chicory, modified inulin is also resulting in a smaller amount of gas formation, which is often felt as a discomfort by humans. And then on the other hand, we are very much interested in the effects of in the gut microbiome. So there is a huge amount of different types of bacteria with different ratios in our gut, and we want to understand how these different samples affect to the ratios of these microbes, and there is a lot to be known still about the overall health effects of our gut microbiome, and we are also working with that aspect in this project. So thank you very much. So this was briefly the bioactivity toolbox that we are working with these interesting modified chicory plants.
Thank you. Our second and last presenter is Dr. Johanna Hoffman-Dietrich. She's an artist, an academic, and a professor at the University of Fine Arts in Poznan, and a fellow of the Ministry of Culture and National Heritage in Poland. Hoffman-Dietrich mostly works in the field of multimedia installation, experimental video animation, and book art. She's also co-founder and chair of the Art and Science Node in Berlin, Germany, and also chair of the Club for Science and Art in Poznan, Poland.
We know today that our world is a dynamic net of interconnections, a dynamic self-organizing system of interactive data flow, from neural networks to the World Wide Web, from river backwaters to social, economic, and cultural phenomena. Our healthy future depends on this network, on plants, on agriculture, soil management, on efficient distribution of resources, goods, and knowledge. The healthy future of our global society cannot be based on famine, malnutrition, or obesity. Can we follow the network solutions mastered by plants to build long-term relationships and provide optimal living conditions for all? Today, I would like to invite you to reflect on these questions by following me on artistic path. I would like to present to you ISN virtual exhibition, Capture the Futures, Our Biotech Planet, The Roots to Roots Networks and Beyond, which had its premiere at the Plant Biology Europe Congress in June this year. Organized by Art and Science Node, the exhibition bridges two interdisciplinary projects. Art Research Education Project Rhizosphere, the Big Network of Small Worlds, and ISN Artist in Residence program by EU CHIC Consortium. Restrictions of COVID-19 concerning physical display motivated us to search for novel solutions. Thus, the exhibition took the form of an original interactive website and an innovative AR augmented reality application. The digital model of a root, devoid of a hierarchical order and a specific direction of growth, is the interface of the exhibition. In the AR version, we can place it in any chosen space. The virtual root has interactive nodules, each of which opens a different world of artistic experience. The website, on the other hand, offers to enter the root interface and discover artistic narratives hidden in its maze. In nature, nodules plays a very important role. They are organs of symbiosis between plants and bacteria, rhizobia, which give plants nitrogen in exchange for carbon. These formations are part of a larger system of rhizosphere, the underground root rhizome fungal networks. It is there that the exchange of resources and signals takes place. These networks, based on symbiosis, diversity, and interdependence, enable plants to develop and spread, and to survive even in very unfavorable conditions. The rhizosphere operating system is based on the Small Worlds network model. Its effectiveness, measured in thousands of years, inspires the search for organizational solutions and strategies that would favor sustainable development and more efficient distribution of resources, information, and knowledge. And of course, it inspires artists' imagination. As an artist, I would like shortly to present two of my works included in the exhibition. I will start with Rhizome Papilla, a project developed in VR and AR technologies in cooperation with composer Andrzej Bartecki and programmer Arek Rataj. Rhizome Papilla is an imaginative organ of new cyber symbiosis, inspired by rhizosphere and symbiotic natural systems of World Wide Web. I wonder how our world, our identity, may change where organic and inorganic systems merge into a new hybrid form of data processing, when the Internet of Things turns into the Internet of Minds. The piece immerses the viewer in a complex, poetic, multi-layered information network in which the boundaries between micro and macro, between internal and external data processing are blurred. The title refers to nodular formations associated with senses of touch, taste, or smell, and to root organs of symbiosis between plants and bacteria. You may imagine the whole world as a rhizosphere, the big network of small worlds based on diversity, symbiosis, and interdependence. Our smallest actions, even thoughts, can affect its well-being. The other piece is a performative poem of Vitruvian woman. It directs the viewer's imagination towards a holistic conscious being that connects our evolutionary past, present, and future. Made in AR technology, the work physically engages the viewer in the process of reading the audiovisual content. The idea of Vitruvian woman refers to the Renaissance tradition of humanists who were exploring the relationship between man and nature. And it draws attention to the similarities and differences between the cultural revolution of the Italian Settecento, symbolized by Leonardo, and of our troubled times, often referred to as the New Renaissance, and bringing together artists and scientists, as we are here today. There is no time in this short presentation to talk about all artworks included in this exhibition and all fascinating and inspiring artistic narratives grouped in 4 areas: hybrid realities, living systems, tracing links, and shifting horizons. I would like very much to invite you to ISN website, where you find the link to the exhibition and to AR apps and also accompanying events. I would like to take also here the opportunity, before we will move to further discussion about crucial scientific societal issues related to the future of food and our global well-being, to thank all artists who participate in the exhibition, as well as to the whole ISN team and especially curator Karina Vlazo-Malinowska.
Thank you so much. We will now meet all together for a discussion about the future of food and medicine. Well, my first question is about the fact that we, as a human species, are now faced with a big problem to meet our future needs for healthy food and medicine. Could new breeding plant techniques, in your opinion, be valuable methods of medical and food production in the future?
Maybe I can start. So indeed, I think new plant breeding techniques can add to to face the challenges that we are foreseeing now. Because with these new plant breeding techniques, we can in a very fast way, we can make at a specific position small adaptations to plants, to the plant genome. And in that way, like what we are doing within our EU CHIC project, we can improve, for instance, the quality. We are working on food fibers within chicory, and we can improve the quality of these food fibers to make them more health beneficial for consumers. So this is a very, very targeted, fast way that is much faster than, for instance, the classical breeding techniques that are used, or even the classical mutagenesis techniques that are used. So I do think that these new plant breeding techniques are very beneficial and can help us to make plants even more healthy, or increase the production of healthy compounds that we can find in plants.
Let's thank you. What about you, Katja? What do you think?
Well, I think that we realize more and more that the plants are used not only for nutrition, but they have functional ingredients that really influence our health. And one of these functional ingredients are secondary metabolites that the plants are making. So all these small molecules that are used as antioxidants or that are even used as medicines. And even there we can use new plant breeding techniques to make more of those compounds or to study how the plants are making these compounds. And I think we have a great need not only for making enough food, but also to make healthier foods. And that is going to contribute to the future of the food, and also not only to the long life, but to a long, healthy life that we need to lead.
And Suvi, do you want to add to what the two others have said?
Yeah, I can only agree with Ingrid and Katja that definitely the plants are huge reservoir of of interesting and bioactive compounds, and there is so much that we still need to understand more about plants. And overall, in the future, we need to better valorize the waste streams and byproducts of different industrial streams, and understand how to utilize the biomass, which may actually contain a lot of valuable compounds, and they can be, for example, used for the medicinal purposes or pharmaceuticals and or different types of applications. And many of the compounds, as Katja mentioned, are very rich in bioactivities. And for example, we know that the huge global threat of the antibiotic resistance is really something that we scientists are trying to tackle, and that is also something that we can find from various types of plants, some kind of solutions to that problem.
Yeah, it's interesting how important it is and how rich the chicory plant is in relation to the future of food and medicine. I would like now to ask Johanna if she has a question.
I think that more than ever, our media art collaborations within the biotechnological sciences are essential to provide provocation, debate, and raise public awareness about the future of food. I have a question to you. How important is to open this debate about the future of the food to the public and communicate research in novel new ways?
Maybe I can comment on that. I think as scientists, we are very much used to exact communication. We like to describe our findings, our results of our experiments in a very exact language. But what I found it a really inspiring experience to now collaborate with artists and look at the science from a new perspective. For example, Jill and Marile, they asked me how the molecules that we work with would sound. So they could hear a molecular orchestra in the beautiful compounds that are there in chicory. Or, for example, we had a cooking workshop working with healthy ingredients from chicory, and then again put chicory in the focus and talk about it from that perspective. So what happens in our body when we are eating a plant? And I think that's really inspiring, and it also inspires curiosity. And it's another way of, again, talking about science, but in a really more inspiring and curious way. So for me, it's really a nice experience to actually talk about science in a new fashion, in a new way.
And I think that what we can add is a lot of interactive potentials so that the public learns how to, about this whole compound structures and deeper ideas about what scientific research is going on through an interactive and media art process, rather than through the traditional processes of painting and drawing, but much more about hands-on kind of tacit learning experiences. So that makes a big difference. What do you think, Marilla?
Well, I think as a filmmaker, I'm kind of in the position of the public, and the valuable participation in the Chicory project was that I could meet you and I could ask you, and you were so open to us, to Jill and me, to explain, and then from our side we tried to do the best to translate your answers to a public.
Do the others have something to say about it from Sylvie or Ingrid?
Maybe I can add that in the past, scientists didn't do this enough, I think, to communicate about the work that they were doing. And especially when we are now talking about the future of food, which is so important to everyone, where scientists also can add to improve our food. There, I think it's even more important to communicate and to have the debate and to explain via, for instance, the artist, the things that we are doing, because this communication with consumers is extremely important.
Yes, and I think we sort of study what we call the semiotics of communication in order to help bring science to the public, but it's very interpretive and not necessarily factual based.
I agree that of course this communication cannot be emphasized too much. So I think that it's really important, and especially when we are talking about these kind of new plant breeding techniques. And then I think that the information flow to general public is also extremely important, and to understand what we scientists are doing really, and what are the concerns from the other side also. So we need to have good interaction, and this art and science project is really helping in that.
I think that actually art is a fantastic, great tool to communicate complex issues.
Mhm.
Thank you so much. I think we're going to stop there. Thanks so much for participating and collaborating. And we would like to especially thank the Art Science Node and the CHIC EU 2020 Horizon Project for their involvement in this seminar. We also thank Laser Leonardo and Ars Electronica 2021 for creating such an interesting opportunity for us to show what we're collaborating about. Thank you.